Hello everyone, it's Dr. Anna Zelaya again with another tutorial. This time we will be continuing with our analysis we're going to be using Ranacapa today for community analysis. And so this is going to be, there's also a handwritten tutorial, and so this is going to be tutorial number 5 of the handwritten tutorials. So number 3 was using DNA subway. And so then you should have completed that by now. Then number 4 was preparing your files to continue with downstream analysis. So hopefully you've done that one. And then now this is one of the options of downstream analysis, which is Ranacapa. And so this is tutorial number 5. So definitely feel free to follow along with the written tutorial and follow along here on the live video tutorial. Okay, so to get started, log into open up a browser. Chrome is always better if you are on a Mac or any other PC. If you have Chrome, those tend to work better than Safari, which is the default browser for Macs. So open up a browser and then visit this website here, gauravsk.shinyapps.io/Ranacapa, and then another backslash, and that'll take you to this homepage. So like the DNA subway tutorial, I'm going to try to be as brief as possible with this tutorial. Although there are some additional concepts that we will have to cover so that you'll be able to interpret the graphs and the results of the community analysis. So this might be a bit more involved than the DNA subway tutorial, but I'll try to keep it brief. There are bound to be questions when learning this new material, so please email me. Again, my email address is anna.zelaya@emory.edu. I'm always happy to answer any questions you have. So this is the welcome page. We're going to go ahead and go to this data import tab. And this is, we're going to basically import our 2 files that we prepared in tutorial number 4. So this here has a demo, so you can walk through all of these tabs with a demo data set that they have pre-uploaded, but we are just going to go ahead and go to custom here to be able to import our own data. So the first one, it says, please select your taxonomy table. It should be either a biome or text file. So in tutorial number 4, we prepared our level 5 CSV file. We prepared it in a special way so that it could be imported here into Ranacapa. We saved it as a .text file, and we labeled it level 5 transposed Ranacapa, if you remember that. So we're going to go ahead and upload it here. We're going to hit browse, and then it's going to open up another window here so I can navigate to find my file. Mine is saved on my desktop in this folder called Bean Beetles. And I have basically all of my files here for the Bean Beetle project. So the taxonomy file was this one here. We labeled it level 5 transposed Ranacapa, and we labeled it as a text file. So it says here plain text. So we're going to hit and click on that and then open it, and that's going to import it here. And then you see it here that it actually like uploads it for us, and you can see all the contents of the file, which is really nice. Then we need to also upload our metadata file, which if you remember back from, I believe it was tutorial number 1, we created 2 metadata files. One was for DNA subway, and the other one was for Ranacapa. So that's what we're gonna upload here is our metadata file, and it's gonna be metadata number 2 is the one we're gonna use for Ranacapa. And we're gonna upload that, and then you see it down here on the bottom. There's our metadata file. And so then we're gonna go ahead and run the app, and hopefully there will not be any errors when we run the app. So we click the app, and then it says, congratulations, no errors detected. If for some reason you have red letters there that have some kind of error warning, it probably means that there's some problem with the formatting of either the taxonomy file or the metadata file. And so if that happens, just go back to the tutorial For the metadata file, it was tutorial number 1, and for the taxonomy file, it was tutorial number 4. Go back and make sure that all the formatting is correct. Ranacapa requires that the files have very specific things in them, and so if Ranacapa doesn't find them, it gets unhappy and it says that it can't read the files. So just go back, make sure that everything is formatted properly, and then come back to this and re-upload it into this data import tab, and hopefully you will eventually see this congrats, no errors detected. Okay, so we've uploaded our files, and now we're gonna go to the next step, which is sequence depth. Now, the above text on this tab has a really nice description, a really brief, nice description about sequencing depth and rarefaction and the importance of all of that for community analysis. If you remember in our DNA subway tutorial, there was a little bit that we covered about that, but I also mentioned that we would talk about it more in depth here in the Ranacapa tutorial. And so that's what I want to do now. So before we get in depth on that particular tab, I want to just kind of introduce a few terms so that it will help us better understand the idea about rarefaction and sequencing depth. So let's pretend that we have a community. So we're going to call this community 1. And as you can see in this community, this is a forest community, and so it has some trees in it. And there's a total of 10 trees in this community. So we could say that the sample size of this community is 10. It's the total number of trees in this community. There are 3 unique types of trees, though. 3 unique species of trees. We have pine trees, we have palm trees, and we have oak trees. And so even though we have, the number of each of the trees doesn't really matter when we're talking about richness. Richness really just means how many unique kinds of an organism, in this example, trees, do we have. And so here we have 3 unique types of trees for a total richness of 3. Now, if we compare this community number 1 to a second forest community, and we start to look at, now let's take a look at the second community. So this community here also has pine trees and oak trees and palm trees, but you'll notice we also have different kinds of trees in addition to that. We have spruce trees, we have birch trees, and we have willow trees. And so in this community, we have a total of 30 trees. So our sample size for this community is 30. But our richness, the types of unique trees we have, is 6, because we have 6 unique types of trees. So if we were going to make a comparison now, if we were going to compare the diversity of community number 1 and community number 2, and when we're talking about diversity, we're talking about the richness or the type of unique the type of unique trees that we have between these 2 communities. On the surface, it looks like, well, community number 2 has higher richness because there's 6. And community 1 only has 3 unique types of trees. But some ecologists would say that this is an unfair comparison because community number 2 has so many more total trees than community number 1. Community number 2 has 30 trees, almost 3 times as many trees as community number 1. So some ecologists would say it's not fair to say that community number 2 is more diverse or has a greater number of unique trees because in total they have so many more trees that it's basically just a numbers game at that point. There's more trees, so by default you're going to have a greater, you're going to be able to find more unique types of trees. So this is where the concept of rarefaction comes in. And in rarefaction, we're basically asking if we had 2 communities with the exact same total number of trees, what would we expect their richness to be? So if I then took a random subsampling of 10 trees from community number 2 so that the number of trees in community number 2 would match those of community number 1, I just randomly picked out 10 trees from the original 30, what unique trees would I see? And so in this example, I basically randomly, well, this was a PowerPoint, so I randomly deleted 20 trees without even looking. And that's kind of equivalent to a random subsampling of data. And then this is what we get. This is the result. Now, in this random sum sampling, I still got a richness of 6. So in this particular case, richness didn't change. Now, if I repeated this process and I again started with the original community of 30 trees, and I randomly subsampled 10 trees from this original 30, I might get a different result. And in fact, that's what happens here. So in this case, I went again and did another round and randomly deleted 20 trees without looking so that I was only left with 10 trees. And you'll notice that the willow trees, I completely, those are gone. And so now we only have 5 trees. Now, the willow trees, there were only 2 of those trees in our original community. And so typically, organisms that are found in very, very low numbers are more susceptible to being removed completely during a rarefaction process. And so that's why here now we see our richness went down. Now, if we were to sample this, do this process again over and over, do random sum samplings like this, the richness will, sometimes it'll go down, sometimes it'll stay the same, but eventually you start to get an average, I should say an average expected richness. And so essentially that is what this rarefaction step is doing. It's taking random subsamplings, you determine the number of subsamples that will be taken, and an expected richness is calculated and plotted like this in what's called a taxon accumulation curve. And so please read through the content. There's also optional reading here, which is it's really good reading to get a really good sense of what the importance of sequencing and rarefaction is to community analysis. And so if we now go back to our community data sample, what we're seeing here is when we say the sequence sample size, essentially what that means is that, so each one of the ADZUKI3, ADZU4, BEP3, BEP4, these are all different samples. These are all different communities. So community of ADZU3, community of ADZU4, community of BEP3, etc. So we have essentially 6 samples or 6 communities that we're comparing here. The sequence sample size means how many total sequences were in any given sample. So if we go back to our tree example, our tree communities, this would be the total number of trees. So in our tree community sample, community number 1 had a total of 10 trees, and community number 2 had a total of 30 trees. And so that's essentially what's being shown here. It's the total number of sequences that were found in each one of these samples. Now you'll notice that this plot is plotting an unrarefied sample taxon accumulation curve. So these are not rarefied. These are all showing the original sample size of all of the samples, and then basically showing the richness in these samples based on their original sample size. So now down here, these graphs down here, this is now the graphs showing the rarefied samples or the rarefied data. And so the number that we would like to rarefy to is selected up here. And by default, it always sets to the smallest sample size of your samples. So in this data set, our smallest sample had 90,164 sequences in it. So by default, that's the lowest that we can rarefy. When we rarefy to that value, this is the results that we see down here. So each sample is basically plotted independently, and it shows what the expected species or the expected bacterial richness would be when we randomly subsample up to 90,164 sequences. Now, what you notice is that these curves all eventually start to plateau. This is what we want to see in rarefaction curves typically. So what that means is that, let's start by showing what this is plotting. So we have, at this point in the plot, at 0, the richness here is also 0. And that makes sense. If we sample one sequence, or I'm sorry, if we sample 0 sequences, we don't have anything to count. So the richness or the species that we see is 0. If we take one sequence out, that one sequence is probably going to belong to a species. And so then the number of richness that we see there is 1. And 2, 3, 4, etc. As you keep pulling out new sequences, you'll start to count more and more unique types of bacteria. But eventually, we get to this point, and at this point, it's at about maybe 20,000. Let's be generous and say 25,000. At about 25,000, what the software has counted as unique bacteria is about, let's say, 30. And so what that means is that even though it's randomly subsampled 25,000 sequences, it's only found 30 unique bacteria in those 25,000. And when it starts to plateau like this, it basically says it doesn't matter how many more sequences the software samples, it's not finding anything new. So the 30 that were found, that's it. That's all that was in the sample, because the more and more sequences that it subsamples, it's not finding anything new. And that's good. That means that we adequately sampled this community. That means that the number of sequences that were obtained was a good representation, allowed for a good representation of diversity. If this line kept going up and up and up and up and up, and it never plateaued, what that would mean is that even if we sampled at 90,000 sequences, the software is still finding new bacteria, new types of bacteria every time, new unique types of bacteria every time it samples more and more sequences. And that means that we wouldn't be able to be confident that we can accurately determine the diversity of this sample, because the software keeps finding new bacteria, and 90,000 sequences isn't enough to truly estimate the diversity of that sample. That's what that would mean. But we don't see that in any of these samples. They all plateau. So that's really good. That's what we want to see, so that we can have a fairly accurate assessment of diversity across our samples. And so what we see now is that we see some samples that had very low richness. So Adzuki here had about plateaus, maybe around 20 unique bacterial types. We have this black-eyed pea sample here that plateaus really high. It plateaus at about 60. And so that's the sample with the highest richness of all our samples. And so these are for your reports. When you put this data into your reports, these are the kinds of observations that you want to be talking about in your reports. Which samples had low expected richness, which samples had high expected richness. Now you can also play with these settings here. For example, you can select a higher value of rarefaction, you can change this number and adjust it. So let's say if I were to put it at, let's say I put it at the maximum number. So what I'm saying now is I want the software to rarefy samples, but I want it to randomly select 136,000 sequences. And watch what happens. If we go down here, all our samples went away. And the only sample that we see here is this BEP number 4. And what that means is basically the software couldn't perform this function because these other samples didn't have 136 sequences in them to randomly subsample. They were smaller in sample size. And so the software just physically couldn't do it. If we adjust this, let's say to 100,000, now we see 4 samples here. So that means that these 4 samples had at least 100,000 sequences so that it could rarefy and randomly subsample 100,000 sequences. But our Adzuki samples are gone because these were our smaller sample sizes. So if you were to do that, you wouldn't be able to compare all of your samples. So that might be a little advanced. Maybe that's not necessary for what you're doing for your analyses, but I just wanted to show you that that is an option that you can kind of change these settings. You can subsample. In our tree example, I talked about if I were to randomly rarefy or randomly subsample many different times, and then you start to get an average. So you can kind of play with that too and random more and more and more times and see if that changes anything here. But again, that's something, just the basic analysis, you can just leave it at these default settings and then kind of try to interpret your results that way. Once you're done with this tab and you've explored all the different options that you can do, you might want to save these graphs for your reports because these are really nice graphs to put into a written report. And so what you can do here is if you click on the graph, you start to see these little icons here. And so you can download the plot, you can try to zoom, you can have all these different options here, so you can play around with that. But I would definitely recommend downloading the plots, both the rarefied ones and the unrarefied ones, so that you can talk about them in your report. Another option you can do, which is also nice, is you can select the variable here. So here I'm putting host, which was the different bean types that we fed. For you, it might be the treatment, the experimental treatment that you did for your samples. And then it'll kind of group them in color-coded. It's just kind of nice. It reduces the number of graphs, and it kind of just helps everything be more succinct. It also gives you some good information about the reproducibility of your replicates. So in this particular experiment, we had 2 replicates of each bean type. We had 2 Adzukis, 2 black-eyed peas, and 2 hyacinth bean samples that we sequenced. Here, the Adzuki beans were actually highly reproducible. As you can see, the 2 samples, their graphs are almost identical. But the black-eyed peas, those were not reproducible samples. So we had one sample that had really high richness and another one that was almost half of the richness of the other one. And the same for hyacinth. And so this gives you some good information about the reproducibility of your replicates. And so looking at my data, I would say that maybe 2 replicates for black-eyed peas and hyacinth, maybe that's not enough. If I were to redo the experiment, to get a better, more accurate determination of richness for those bean types, I might want to add more samples because there's going to be a lot of variability. Some might look high and some might look low, and that's not really a good or accurate assessment or estimate of that diversity. So once you're done with exploring this tab and downloading all of your plots, you can move on to the next tab, which is the taxonomy bar plot tab. Okay, so in this next tab, the taxonomy bar plot tab, This page allows you to visually compare the relative abundances of all of the different taxa across all of your samples. And so what you see here is essentially a stacked bar plot. We have each sample along the x-axis and the total abundance of, basically, abundance here means the number of sequences that were found in each sample. You'll notice here that the unrarefied tab is checked right now. So this tab is showing us essentially all of the sequences found in each sample. So you'll see here, if you remember correctly, or if you remember from previous, our Adzuki samples were our smallest samples with about 90,000 sequences. And we had this BEP sample that was the largest. I believe it had like 160,000 or something like that sequences. So that's what we're seeing here essentially. And then each different color represents a different bacterial taxa, and it represents how many sequences of that taxa are found in a sample. Right now, the phylum category is chosen, which means that all of these colors are representing the different sequences that have been classified at the phylum level. And it's the abundances of that phylum. So we have here, if I click on this pink section of the part plot here, we see that for this Adzuki sample, the pink represents the phylum Proteobacteria, and there's about 76,000 sequences of Proteobacteria in this sample. And we can do that for all of the samples. So pink will be Proteobacteria in all of our samples. And the value here is 77,000. We have some pink in this sample, but it's very, very low abundance. It's only about 1,500. And we have some as well here at about 6,900. And then here, it looks like we do have some, but it's so low that you can barely see it on the graph. So here we only have about 300 sequences in this sample were classified as proteobacteria. So that's how you read this graph. I want to just briefly cover just the taxonomic levels in general. And so here I'm putting up a photo of the taxonomic classification of a dog. And taxonomy or taxonomic classifications are the way that ecologists and biologists have grouped living organisms. In modern taxonomic classifications, organisms are typically based on their genetic similarities. So for our data here, we sequenced a part of the DNA of bacteria, and part of what we did in DNA Subway was to then group those sequences that were similar to each other and then reference them to a database that would tell us how similar they are to other organisms. And then that's how we get that kind of grouping or similarity and classification of different organisms. So example, if we were to sequence dog DNA, we could then make kind of a classification like this, where we could group it based on other things that it's genetically related to. And the way that we group them can be at different levels. So the furthest, most distantly related would be at the domain level. So in our example here for dogs are in the domain Eukarya, and that means, genetically speaking, they are genetically similar to all of these other organisms that you see here, plants, insects, fish. And if we look at less distantly related, we can see the kingdom that is classified under, and all the other different organisms that are classified as the same kingdom. And so they're genetically related because they're all falling within the same kingdom. And then phylum, class, order, family, genus, species, until we get to dogs, which all dogs are this particular subspecies. So you can do this for all living organisms, and you can do this for bacteria as well. And so bacteria can be classified by their domain, their domain is the domain bacteria, and their phylum, their class, their order, their family, and how related they are to other bacteria And so for our purposes, so you can play with this here and you can look at different, you can look at how the different bacteria were classified when it comes to class, order, family, etc. We will go down to family. We won't go down to genus or species. And the reason for that is the the length of DNA that we sequenced and compared is a relatively short piece. And so if it's too short, it's difficult to classify organisms at the genus and species level. And so that's why we're just gonna go to the family level. We can do phylum, class, order, family, and then we won't go any further. So if we look at the family level, and you can do this for your data set as well, you can then compare the different samples and how basically the types of bacteria that are found in each sample. And you can make some comparisons about your samples. So for your report, some things that you might want to mention and talk about is you want to compare your different samples, compare the different taxonomic levels, and compare, you could compare how similar your replicates are. So in this example here, my two Adzuki replicates are very similar. We have Enterobacteriaceae, Staphylococcaceae, and some sequences that were not able to be classified at the family level. So it says unknown. That's fairly common. So you might have some of those as well in your sample data set or in your data set. But essentially what we see is we see the same three main types in both of my Adzukis, and they're fairly similarly abundance as well. The abundance is quite similar across both. If I look at my black-eyed peas, again, we're dominated here by Staphylococcus. That's the most of the sequences were classified as Staphylococcus. We do see some Enterobacteriaceae in this black-eyed pea sample, but less in this one, much less in this one. So there's our, you can barely even see it. It just kind of looks like a big black bar because there's other bacterial types all clustered really closely together, so you can't really differentiate their colors. But you see this sample has about 900 sequences versus this sample that has about 6,000. So there's a lot more in this particular sample than there is in this one. And so these are the kinds of things you want to do across all of your samples. You want to compare your two different replicates. You want to compare across types. So I can compare how different the Adzuki microbiome is compared to the black-eyed pea microbiome, et cetera, in terms of the types of bacteria that are observed and their abundance. Another thing you will notice, I think I mentioned it already, that this is the unrarefied abundance. You can also look at the rarefied, which I would recommend you do, because again, as we talked about in our previous tab, a rarefied data set is something maybe you can, it's a little bit easier to compare because you're comparing the same number of sequences. And so again, here we see a similar thing where we see the same kind of amounts between our Adzuki beans of the three different types. And we see a slight change, which is expected for rarefaction with our black-eyed peas, but in general the trend is similar. They're still both dominated by Staphylococcus. So go ahead and do that for your samples. Look at the unrarefied, rarefied, and then go through the different colors, how abundant they are, what's the most abundant in each of the different sample types, how similar are your replicates, and things like that. This is what you'll want to do for this particular tab at the family level. And then you can download this plot by clicking up on here, which I would definitely recommend you do because it's a nice graph that you can include in your report. So after you're done with this tab, exploring all the different options and making all the different comparisons, we can move on now to the next tab, which is the taxonomy heat map. And the taxonomy heat map is really, it's essentially the exact same thing as the taxonomy bar plot. So I won't spend too much time on this one. It's just a different way to look at the taxonomy data. The first tab was a bar plot format. This is a heat map format, but the information is exactly the same. It just looks a little different. So all of these, we have here all the list of all of the different bacteria classified at the family level. Our different samples are along this x-axis. And you see here now they're kind of color coded based on this legend here. So we have these kind of purple, darker colors represent more lowly abundant organisms. And these brighter yellow colors represent what's more highly abundant. This is kind of nice because you see there actually was a lot of different bacteria in our samples. It wasn't just, 2 or 3 like what we could see in the bar plot. We're only really seeing 2 or 3 main colors. And so it makes you think, oh, there's only 3 bacteria here. At the family level, there's only 3 types of bacteria, but actually there's a lot more. They're just all very, very lowly abundant. There's only a few sequences of each of these different families. So it's they're a little bit more hidden in the other one. But essentially it's showing the same information. We see here Enterobacteriaceae is the most abundant in Adzuki. The Staphylococcaceae is the most abundant in black-eyed peas. We have this, again, Staphylococcus are the most abundant in hyacinth, which is what we saw in the other plot as well. And again, you can do it for a rarefied or unrarefied, and you can do it at the different taxonomic levels. So however it is that you'd like to look at the data, so similar but different. So this is all basically just preference at this point. How would you prefer to show your data? Some people might prefer a heat map where you can see a lot more of the different types of organisms there are. Somebody might prefer a bar plot because it's showing the most abundant ones. So that's all preference at this point. Again, you can download this graph if you'd like to include it in your report. So once you're done looking at this tab, we can go to alpha diversity plots.
Okay, so in this tab, we're going to be introducing the concept of alpha diversity. So alpha diversity essentially just means we're trying to assess the diversity within a single community. And Ranacapa discusses alpha diversity in terms of 2 measures, observed richness or observed diversity and the Shannon diversity. And to explain these concepts, we will go back to our forest example. So going back to our forest community, we are looking at a rarefied version of our community. So both communities have the same sample size of 10 total trees. And when we count up the unique types of trees in each community, we see that community 1 has a richness of 3. And our second community has 6 unique types of trees, or has a richness of 6. So based on richness alone, or observed richness alone, we would say that community 2 has a higher observed diversity or higher observed richness than community 1. But another way to look at alpha diversity is to also take into account how many of each tree there are in these communities. So that's when we would then count the abundance of each tree. And so let's take a look at community number 1. Community number 1 has 8 pine trees. It has 1 palm tree and 1 oak tree. And so looking at that data alone, we can kind of already see that this community is very heavily dominated by pine trees. 80% of this community is composed of pine trees. Pine trees are very largely represented in this community, and there's very little of any other tree. Now, if we look at community number 2 and count the abundances of each tree, we see that there's 1 pine tree, 2 palm trees, 3 oak trees, 1 spruce tree, 2 birch trees, and 1 willow tree. And so if we convert that into percentages, we see that each tree is about 10 to 20% represented. Oak trees are the most represented at 30%, but they're all kind of at 10, 20, and 30%. So if we look at that data alone, we can determine that there really isn't one type of tree that's dominating this community. They're all fairly evenly represented. So the Shannon diversity measure is um, we calculate that measure with an equation that you don't need to know, but just know that the equation takes into account the richness of the community, the abundance of each of the organisms in the community, and the sample size of that community. And it takes all of that information and it calculates a Shannon diversity value. Now, the calculated Shannon diversity value for community number one, I'm not going to go through the calculation, but I would expect that the Shannon diversity calculation would be lower. It'd be a lower value for community number one than it would be for community number two. And that's because it's also taking abundances of each organism type into account. And it's really looking at how diverse really is the community. Is it dominated by one type of organism? If it's dominated by one type of organism, then that's not really a truly diverse community. But for community number two, that's not necessarily the case. We have a lot of different kinds of trees, and they're all fairly equally represented, and that would all contribute to a higher Shannon diversity. So if we go back to the alpha diversity tab on Ranacapa, we see that the first chart here is showing us the observed diversity, which we learned previously that basically is the same thing as richness. So we're looking at the observed richness, we're calling it now observed diversity for each of the samples. And that's the first way to look at alpha diversity on this tab. The second way that we can look at alpha diversity is by looking at the Shannon diversity. And so here, this bar plot, the Ranacapa software calculates Shannon diversity for us, and then it shows us what that, or it plots those values on this chart here. And now that the values are plotted, we can compare samples that might have higher Shannon diversity than other samples, which samples have higher Shannon diversity compared to other samples that might have lower Shannon diversity. And so let's go ahead and walk through this sample data set right here.
By clicking on this, the software calculated Shannon diversity for us. And what we see here is that when we calculate Shannon diversity, our hyacinth samples have very high Shannon diversity, and our black-eyed pea samples have very low Shannon diversity. Now let's compare that with the observed richness, which was the total number of unique types of bacteria that are found. We kind of see an opposite result. In black-eyed peas, we have very, very high richness, and in hyacinth, we have kind of lower richness. But when we calculate the Shannon diversity, they kind of flip, and the hyacinth is higher and the black-eyed pea is lower. It's even lower than Atsuki. Atsuki in our previous example was the lowest of everything, but now black-eyed pea is the lowest. Okay, so what does this mean?
Well, if we think back to our forest example, basically what this is indicating is that the black-eyed pea samples, although they might have a lot of different kinds of bacteria, they are probably really highly dominated by just one type of bacteria. And probably hyacinth, that even though they may have less total unique types of bacteria, maybe their numbers are a little bit more evenly distributed as compared to the black-eyed pea. So let's take a look at the taxonomy bar plots and see if that's the case.
And I'm going to use the rarefied example here. And let's go down to the family level. So black-eyed peas, even
So
though they may
that
have
seems
had
to be
the
the case for
highest
us
total
here.
richness of everything, of all of our different samples, they're very highly dominated by one type of bacteria, this Staphylococcus. It makes up almost 100% of these samples. I mean, this entire bar is colored pink. So these samples are highly dominated by just one bacterial type. If we look at hyacinth, which had our highest Shannon diversity, we see that it is also highly dominated by one type of bacteria, but we also see a lot of this second bacterial type, this Enterococcaceae, and we see a lot more of this orange unknown representation, a lot more so. So even though it is still fairly dominated by one organism, it's not as highly dominated as this one, and there's a little more room for different organisms. These green bars are there's a little bit more of them, and there's a little bit more of this orange.
So when we're comparing the diversity of these two samples, hyacinth and BEP, hyacinth gets calculated as being more diverse than the BEP sample because it's not as highly dominated by a single organism as is black-eyed pea samples. So going back to the plots here, basically that's what we're showing here, the Shannon diversities as compared to the different, comparing the different Shannon diversities across the different samples. And if you click on there, you can see the actual value that the Shannon diversity was calculated at, and you can see how they compare.
If you would like a more in-depth explanation about alpha diversities and Shannon diversity, you can definitely check out these links here. They're really good resources to kind of get more information and maybe go into that a little bit more in depth. But again, for your report, you will want to download these photos. I would recommend downloading both the Shannon and the observed. I would also recommend you group them to your treatment type. So here I'm grouping them by the bean diet. So this just makes for kind of a nicer graph. It's more succinct. And so you can, again, do both the observed and the Shannon graphs and download those. Once you're done with this step, we can move on to the next tab, which is the Alpha Diversity Stats tab. This tab, the Alpha Diversity Stats tab, allows us to calculate whether or not the differences in alpha diversity that we just observed in the previous tab are significantly different. And it does this by calculating ANOVAs and Tukey tests. So one thing about, I'm not sure if they've updated this since the last time that we used this, but one caveat that you will want to know is that Ranacapa might not be able to calculate ANOVAs or Tukey tests if you're only comparing two different types of, essentially it would be called two factors. So if you had two different treatment types. So for example, if we were doing this test with just comparing black-eyed peas and adzuki beans, and we didn't have hyacinth beans in our data set, we would not be able to calculate the ANOVA or Tukey tests with Ranacapa. Ranacapa requires 3 or more factors to calculate these values. So just keep that in mind. If your class only has 2 different treatment types, then you might just want to skip this step because there might not be anything that can be calculated for you. But if you do have 3 or more, and also if you have replicates of those, so if you have at least 2 replicates for each treatment type, then you will be able to do these analyses. So it will calculate an ANOVA and a Tukey test for either the observed diversity or the Shannon diversity, whichever you choose. And so it would be good to look at both because it'll calculate whether or not richness is statistically significantly different across your samples, or if Shannon, which takes into account richness and abundance, is statistically significantly different. You'll also probably want to change this to group it for the treatment type, because that's how we will want for these tests to be calculated. So as you can see here for our results, the first one is the ANOVA, and according to this p-value here, it says that our samples, our bean types, the microbiomes that we observed from different bean types were not statistically significantly different in observed richness. That's what this says here. For the post hoc test, for the Tukey test, it's basically doing a pairwise comparison of the different bean types. And it's saying here, again, that basically the microbiome that we observed when we are comparing between these two different types, bean types, they're not significantly different when we're just looking at richness across those different groups. Now let's take a look at the Shannon diversity. So in Shannon diversity, we see something different. We see that according to the ANOVA, there is a significant difference across different bean types in terms of the Shannon diversity. And we know that because the p-value here is less than 0.05. And then we see here, we see which of those two pairwise comparisons are different. So which microbiomes are different based on Shannon diversity. And we see here that the microbiome of beetles fed on hyacinth beans, is statistically significantly different than the microbiome of beetles fed on adzuki beans. And we have this p-value here, 0.04, as are the microbiomes of beetles fed on hyacinth beans when compared to those that were fed on BEP, black-eyed pea beans. So this is definitely something you will want to include in your report. I don't believe you can download these tables, so you can take a screenshot, you can write down the values so that you can have this data for your reports. Once you are done with this tab, you can move on to the beta diversity tab, or beta diversity plots. And again, I'm gonna go ahead and group, I'm gonna select the variable here and I'm gonna change it to host. And so we looked at alpha diversity, which as a reminder is basically looking at diversity within a sample. It's taking into account richness and abundance within a given sample, and then we calculate some kind of value to tell us what the diversity is of that single sample. And then you can kind of compare your different samples and you can see what that alpha diversity value looks like across your different samples.
Beta diversity is a slightly different concept, and we're going to return to our tree example to try to understand this concept a little better. So beta diversity is taking two main pieces of information into account. It's asking how many organisms, or in our tree example, trees, do the two communities that are being compared have in common? And are those trees that are found in common found in the same proportions in both communities? So essentially, beta diversity is comparing the entire taxonomic composition of two communities and compares how dissimilar they are in terms of their taxonomic composition. The dissimilarity value is calculated here in Ranacapa. Ranacapa uses the Jacquard dissimilarity and the Bray-Curtis dissimilarity values.
You won't need to know how to calculate these two different dissimilarity measures. Again, if you're doing the Excel tutorial or the Google Sheets tutorial, you will learn that. So we're not going to go over that here. But just know that the Ranacapa is calculating this beta diversity value and it's plotting it. After it calculates that value, it plots it here on what's called an ordination plot. So this is called a PCOA. That's a type of ordination plot. There are many different kinds of ordination plots. And so this is a PCOA. Ordination plots, they can be a little bit confusing to decipher. So in this ordination plot, the points represent our samples. The way that you would read this is when samples are clustered closely together in this two-dimensional plot, they are considered more similar to each other. And when samples are clustered far apart from each other, they're considered less similar to each other or more dissimilar. So here we have these adzuki beans that they're very, very close together. That means that these two samples are very highly similar to each other. we have these two black-eyed peas samples that they're also very highly similar to each other in terms of the taxonomic composition of these samples. And then we have hyacinth beans, which they cluster fairly far apart from each other, are two replicates clustered far apart. So that suggests that taxonomically, the composition of these two samples is they're not very similar. And in fact, this hyacinth sample, the composition of that community actually looks more, it resembles more the community of black-eyed pea because they're clustered more closely together here than it does its own its replicate. And so again, that's kind of just, that's the biggest take home message for how to read ordination plots. If they cluster closely together, that means they are more similar to each other. And if they cluster further apart, they're more dissimilar. Now, the Jacquard dissimilarity index, that dissimilarity value is being calculated, as you can read here, it says it's being calculated simply based on the presence or absence of an organism. So what this says is that the types of bacteria found in Adzuki are very different from those found in hyacinth and are very different from those found in black-eyed pea.
So that's how you would interpret the Jacquard. It's really just looking at the types of organisms found in the community and whether or not two communities share the same types of organisms in common with each other. The Bray-Curtis dissimilarity value also takes into account shared types of organisms between two communities. But it also takes into account, in addition to that, it takes into account what proportions they're found in, and it's asking the question, are they found in similar proportions or in different proportions?
What you'll notice though is that our plot doesn't look that different, which might not be the case for your data. Your data might, the presence absence plot might look one way and the Bray-Curtis plot might look a different way. For our particular data set, they don't look very different. You can download these plots here again with this icon here. Another way to look at beta diversity is through this hierarchical clustering. It's a similar idea in terms of that samples, the taxonomic composition of samples, if they're clustered together in this kind of hierarchical structure, that means they're more similar. And if they're kind of further apart from each other, then they're more dissimilar. So these two graphs essentially provide the same information. It's just two different ways of visualizing the results. So once you're done with this tab, you can move on to the next tab, which is the beta diversity stats, which is similar to the alpha diversity stats. So it's going to take the beta diversity information that we got from the previous plot, and it's going to give us some statistics and as to whether or not these differences that we saw were statistically significantly different. So again, I'm grouping them by host. And you'll go here, you can start off with the first Jacquard dissimilarity. And what you see here is that based on the ANOVA analysis, we have, if you have the 0.05 as your strict p-value cutoff of significance, then you would determine that this is not statistically significant. I would defer to your instructor on that and what they would say. Some people would say that this is maybe marginally significant. Again, I would go ahead and ask your instructor about how they would interpret this value here as to whether or not the 0.05 value is going to be the hard cutoff for significance. And if we then look at the Bray-Curtis, it looks like it's the same value. It didn't change. It's still the same 0.06. So both Jacquard and Bray-Curtis dissimilarities both gave us the same results here for this data set. So go ahead and do that for your data and see what your beta diversity analysis looks like. So that is pretty much the last tab here. This data export tab allows you to kind of download your data tables again, but you don't really need to do that because you already have your data. You don't really need to do this.
So that brings us to the end of our tutorial.
I walked you through the different tabs of Ranacapa analysis. This analysis tool is nice because it allows for kind of basic visualizations. Another way that you could do these is by using R, which is a programming language. But this Ranacapa software just gives us a nice, for beginners who don't necessarily know how to use R, it's a really nice way to perform community analysis without having to use R, without having to use any program languages or coding. You just click buttons and run through your community analysis. So I hope that this tutorial was helpful. Again, I'm sure there's going to be questions. There's a lot of ideas and a lot of concepts being introduced here. So please email me. I'm more than happy to answer all questions that you might have. And so with that, I will end it there. Thank you for watching, and good luck with the rest of your analysis.